Live na.
Okay na dahil, no? Nasa YouTube na ba tayo? Okay
Yes,
na. Yung link na lang palapasend sa GC natin.
Oh, yan.
Para malagay din nila dyan sa chat. Si GC natin naglalagay ng... Ay, wala pa sa si GC. Si JD na lang.
Yan, okay na.
Ayan. Para sa chat dito, tsaka dun sa FB. Tapos, pwede mo na siguro gawin mo ng co-host ni Ma'am Joan tsaka si Ma'am Nadia.
Alright. I-check po yung sa live stream natin kasi sa stop share kasi ako kapag nag-ugat na
Kaya ako mag-check sa live stream.
Okay. Check kung nag-play.
Okay.
Thank you. Also, yung iba po natin co-host, kung merong board member na pumasok, pa-co-host na lang po kasi hindi ko nakikita.
Okay yung sa ano?
Sa YouTube. Yes, thank you.
Okay, admit na?
Let's go.
Oh.
Okay.
So this is a start. I would like to welcome everyone to this second session of the PSDB 2022 webinar series. May I greet you first, a happy, safe, and pleasant, healthy, and good afternoon to everybody. So this is our second session. Our first session is in April 23, and we are live via Zoom, and at the same time also live streamed on YouTube. So I am Dr. Sonia Jacinto, and I am going to be your host for this afternoon. I will be moderating the afternoon session, and I hope I will take care of you very well. So I am the advisor, one of the advisor, and a member of the Council of Elders, together with Dr. Annabel Herrera and Dr. Ahmad Masaheri, and at the same time, Chris Lopez from Ateneo. May I have the next slides, Jordan? So that's me. Okay. So for the first session, I just like to give a very short recap, which we had on the 23rd of April last weekend. We had Dr. Lilia and Fernando of the Institute of Crop Science, College of Agriculture and Food Science, University of the Philippines from Los Baños, talking about nano biosensors for agricultural applications. And then we also had another PhD, Dr. Yefrosina P. Atabay. This time, she is the supervising science research specialist from the Philippine Carabao Center, national headquarters from Muñoz, Nueva Ecija. So she talked about enhancing livestock production through reproduction, research, and innovations. This time, our second session for this webinar series, we're going to have two equally interesting talks as well. The first will be delivered by Dr. Margaret Ann F. Pelayo, and she is from the Nara Institute of Science and Technology from Nara, Japan. So she will be talking all about epigenetic mechanisms, underlying temporal development, transitions to specified floral stem cell fate. And the second talk is going to be given by Mr. Jabriel C. Baluyot, MC, from the Regenerative Biology Research Laboratory of the Institute of Biology, College of Science, University of the Philippines, on benchmarking endocrine disruptors in water samples using transcriptionally activated human cell lines. Okay, so for the first talk, before we go to the first talk, let us be clear about the webinar rules. I'm sure if this is the second time you have attended this webinar series, you are a bit familiar with this, but it bears repeating, no? So please turn off audio and video upon login. And then you type your questions at Slido, and the Q&A facilitator will collate and read the questions to the speaker. You can also use the raise hand button of Zoom if you want to ask a question live. Please wait to be acknowledged by the facilitator before you unmute your microphone. For PSDB members, links to the evaluation form will be posted on the chat box. Please submit the form at the end of each session. Okay, how do you get a certificate? You must be an official member of PSDB registered in the PSDB 2022 webinar series. And you must submit the evaluation form for each session. And you must note that the evaluation forms will open at the end of the session until 11:59 p.m. today. So we would like to thank the sponsors with deep gratitude, Macare, Merck, MedTest, and Phil Anasser for always being there, co-sponsoring our activities. Okay, so thank you so very much to you, dear sponsors. Okay, now so we go to the meat, so to speak, of our session 2. You are looking at the photo of this nice-looking lady, Margaret Ann F. Pelayo, who had a PhD at the NARA Institute of Science and Technology, who will give her talk all in good time, epigenetic mechanism underlying temporal developmental transitions to specify floral stem cell fate. Dr. Margaret Ann Pelayo received her BS Biology degree from Ateneo de Manila University and an MS degree in plant biotechnology from Ghent University and a PhD in biological science from the Nara Institute of Science and Technology in Japan. Her dissertation research focused on the mechanisms regulating floral meristem maintenance and determinancy in Arabidopsis. She also worked as a postdoctoral fellow at the Nara Institute of Science and Technology, doing research related to engineering temporal gene expression in Arabidopsis thaliana floral meristems. She has done research on various aspects of plant development, plant physiology, and molecular biology in institutions like the University of California, Davis, Ghent University, and the International Rice Research Institute. Dr. Pelayo is a member of several professional associations, which include the Japan Society for Plant Physiology, the International Society for Plant Anaerobiosis, and the Crop Science Society of the Philippines. I'm glad to note that our friendship and collaboration with the NARA Institute of Science and Technology is still growing strong since we started in 2015. So may I call on Dr. Margaret Ann F. Pelayo, please, to take over the stage.
Okay. Thank you, Ma'am Sonia, for your very kind introduction. Can you hear me?
I can hear you. We can hear you.
Thank you. All right. Let me just set up my slides. Just a second. Okay. Okay. Just a second. Sorry. Oops. Sorry. Taking time. Yeah. All right. Okay. Okay. All right. Thank you. Thank you again, Ma'am Sonia, for your very kind introduction. It's very nice to meet you online as well as everyone who has joined or is joining us today. It's really a pleasure to be part of the Society's webinar series. Now, a central theme, if not the central theme of my talk today, is time. And the nature of development is always connected to time. To go through changes from one phase or stage to another is one of the basic tenets of developmental biology, and essentially is a function of time. And in this case, even cells, even at the cellular and the molecular level, as they change and develop from one cell type to another, these events are still bound by space and, of course, time. And what we're going to take a look at today is how these temporal or time-bound developmental events are governed or regulated by epigenetic mechanisms. Right. Now, just like what Ma'am Sonia said, I did my PhD at the Nara Institute of Science and Technology in Japan in Professor Toshiro Ito's lab. And in Ito Sensei's lab, we study flower development in the model plant Arabidopsis thaliana. Particularly that moment when floral stem cell fate is irreversibly sealed. And we call this process floral meristem termination. In this case, a floral meristem, or the FM, is made up of floral stem cells from where flowers will emerge. And FM termination is an important process because this is what will lead to proper flower formation. And initially, the FM will be specified into the different floral organs and is eventually terminated. This latter process, or again FM termination, irreversibly seals floral stem cell fate, which begins at about stage 5 of flower development and marks the transition from cell proliferation, a hallmark of meristem cells, to differentiation in favor of organogenesis. And the molecular process underlying this transition is the core concept of my study. For today's talk, it's important to emphasize that flower development goes through stages from the time in which stem cells become the FM until the time when a fully formed flower is produced, as you can see here. And in Arabidopsis, flower development has about 20 stages. The developmental map shown here is until stage 16. And today, the stages we will be mainly concerned about are in the early part of development, particularly from stages 3 to 8, the time when FM termination happens. And in this case, repression of the meristem maintenance and proliferation protein WUSCHEL is essential to FM termination and can be done either directly or indirectly by the C-class homeotic transcription factor AGAMOS or AG. And then the indirect pathway involves AG activation of the transcription factor knuckles and is the scope of my work and what I will be discussing today. And previously, we found out that switching on the gene knuckles is central to FM termination, and that this is an epigenetic process dependent on the cell cycle. So what exactly does this mean? Initially, knuckles is switched off via repressive histone methylation marks, or H3K27ME3, along its coding region, catalyzed and maintained by the polycomb repressive complex 2, or PRC2, bound at the knuckles promoter region. Then at stage 3 of flower development, AG will competitively bind with PRC2, which evicts PRC2 from the knuckles promoter region. However, at this point, knuckles is still switched off. Successive rounds of cell division, approximately 2, is then required to dilute the repressive K27ME3 marks, leading to full knuckles activation after a 2-day delay by floral stage 6. Thus, switching on knuckles is an epigenetic process involving H3K27ME3 dilution or passive K27ME3 removal that is dependent on the cell cycle. Currently, knuckles is the only known gene regulated via this intrinsic timing mechanism, which I will call biotimer mechanism from here onwards, and how chromatin status affects this temporal regulation is still unknown. Thus, these are the questions I ask with my study, particularly, are there other genes regulated similarly as knuckles? And what role does chromatin status play in the biotimer's temporal regulation? And to answer these questions, I performed genome-wide in silico analysis and some implant analysis to identify candidate biotimer genes, and chromatin manipulation at the knuckles coding region, as knuckles is the seminal biotimer gene, to see if I can modify knuckles temporal regulation from a chromatin approach. First, I'll discuss our results in silico and implant analysis. All right. And I propose that the biotimer genes They are defined by their direct AG and PRC2 targets, are enriched for K27ME3 marks, and which show transcriptional delay upon AG and AG binding associated with cell division cycles. Thus, I used 4 publicly available genome-wide datasets that correspond to the biotimer definition, namely AG, namely chip data, for AG and PRC2 binding, as well as K27ME3 enrichment data, then flower stage specific transcriptome data from an AG loss of function line. And I use the following criteria to screen these datasets, namely significant gene expression change in stages 6 to 7 relative to stages 3 to 4, and significant AG and PRC2 binding and H3K27ME3 enrichment. This led to 11 candidate biotimer genes out of about 4,000 included in the screen, which includes knuckles. And 8 out of 11 of these genes are transcription factors. Then the 2 others are a heat shock protein and an oxygenase protein. And most of these genes seem to be flower specific and function mainly in development. Then SPL, here below, was manually excluded from the list because previous findings have shown that SPL is transcribed immediately upon AG binding. Then to learn more about the gene expression pattern and function of these genes, I compared the expression levels between stages 3 to 4 and stages 6 to 7 in the AG loss of function line, and found that 2 of them are upregulated without AG, shoot meristemless or STM, and zinc finger here at the top, and the remaining 6 are downregulated. In this case, red means upregulation and blue means downregulation, suggesting that a functional AG is necessary for transcription and based on biotimer regulation is necessary for the repressive K27ME3 removal. Then further go-to-room analysis and refinement show that the candidate genes are mainly associated with transcription and development, suggesting the existence of a biotimer gene regulatory network downstream of AG. Next, to learn more about how these genes behave in planta, I performed several time course experiments, starting with looking at the gene expression profiles or patterns through real-time qPCR using an AG inducible line, meaning we can time when AG is activated and thus also its downstream targets. And I checked the expression from 0 hour with no AG induction until 7 days. These are alternating profiles after AG induction. And the very first panel is for knuckles, and we see the lag or 2-day delay of knuckles transcription. Its activation or expression doesn't increase significantly until 3 days after induction. That would be panel A. Then the next set of genes, IDD12, AHL18, PLATS10 have similar expression profiles as knuckles, and those at the bottom panel tend to have different expression profiles. In this case, this one, panel E, an oxygenase protein, is induced immediately and its expression also decreases immediately. Then panels F and G, STY1, and a homeodomain live protein, their expression is gradual or gradually increases. Then for HSP20, the final panel, we hardly detect any expression at all. Okay. Then for knuckles and the 3 genes with similar expression profiles, we further check the Phi binding and also K27ME3 accumulation or enrichment, and also cell cycle dependent changes in expression. The first 2, Phi binding and K27ME3 accumulation via chip, show that after 3 days, we also used inducible lines, in this case, the AP1-Cal ones. Phi and K27ME3 marks are depleted. So PRC2 and K27ME3 tend to have a negative correlation with gene expression. Then we also inhibited the cell cycle, here panel K, using olomycin, a cell cycle inhibitor drug. And the premise is that if we inhibit the cell cycle, the genes would also be inhibited. So knuckles and the other candidate genes do show significantly reduced expression after 3 days of OLO treatment relative to samples that were not treated. And we also have a negative control, a gene called CRC or Krabs clause, also an AG target, but not a biotimer candidate, showing its transcription is not affected in the presence of olomycin. Okay. So taking everything together, just a quick summary so far, this suggests that the biotimer mechanism may involve multiple strategies to execute proper temporal activation of the candidate genes. And these strategies may encompass variation in chromatin conformation and environment that could allow for maybe a dynamic response to intracellular and extracellular signals, resulting in proper transcriptional induction timing. Okay. Next. So next, for the second part, I was also interested in the role of chromatin status in the biotimer's mechanistic regulation. And for this, I performed knuckles chromatin manipulation to alter its temporal activation. Right. And for this, a region named DEL in the knuckles coding sequence is especially important. The knuckles coding sequence comprises a 486 base pair region, and the image below shows the wild-type knuckles expression pattern in the developing floral flowers on the Arabidopsis inflorescence, represented by the blue signal you see here on stages 6 to 8 floral buds, and the blue is from the Gus reporter. Previously, it was found that deleting a 231 base pair area downstream of the knuckles coding region, which is highly enriched for the K27ME3 mark and is bound by the PRC2 component phi, causes early and ectopic knuckles expression as seen below. Hence, this 231 base pair area, which was named DEL, stands for deletion, is important for proper knuckles temporal activation. I then took the reverse approach with my current study by adding more of the DEL regions into knuckles. And the main question I am asking now is, can knuckles temporal activation be delayed this way? My hypothesis is that extending the knuckles coding sequence using iterative addition of the DEL region could also be sufficient for the repression of knuckles in a cell cycle dependent manner, wherein chromatin status is a key mechanistic determinant of the biotimer. So first, DEL was added iteratively from 1 DEL to 6 DEL in 2 knuckles to find out systematically how DEL affects knuckles temporal activation. And what is displayed here are the graphical representations of the constructs for my plant materials. Gus was used as the reporter system to find out the knuckles expression pattern when DEL is added. And in this case, the solid black line here represents the region upstream of the knuckles coding sequence. The white bar is the 5' untranslated region or 5' UTR. The black bar and the gray bar is the entire knuckles coding sequence, and the gray bar specifically represents the DEL region. Then the gray bars with the plus sign represent the DEL regions that were added, and the blue bar represents the Gus gene. Then the black line on top of the DEL and Gus junction represents the area for our primers for our qPCR and chip experiments. Then I observed knuckles stage-specific expression pattern from floral stages 4 to 8 with the wild type as a control. The wild type knuckles expression pattern begins at stage 5, peaks at stage 6, then subsequently decreases from stage 7 until stage 8. Then I compare the wild type knuckles expression pattern from stages 4 to 8 with homozygous 3 DEL and 6 DEL lines, and found that as DEL is increased, the knuckles expression pattern also attenuates and is eventually delayed. So again, wild-type knuckles expression begins at stage 5, while in 3 DEL, knuckles is only weakly expressed beginning at stage 5, and this continues until stage 6, peaks at stage 7, and decreases by stage 8. And in 6 DEL, knuckles expression begins at stage 6 instead of stage 5, peaks at stage 7, and is unobservable by stage 8. suggesting that adding more del and knuckles can delay and also decrease knuckles expression. Then, Gus transcript accumulation was quantified by a qPCR using inflorescences containing up to stage 12 floral buds and resulted in a significant decrease in Gus expression levels in 6-del relative to knuckle Gus. Then, quantification of endogenous knuckles expression levels yielded no significant difference between 6-del and knuckle Gus, suggesting that del addition doesn't affect endogenous knuckles expression. And finally, quantification of Gus enzyme activity through MUG assay resulted in a decrease in Gus protein activity in 6-del, consistent with the decreased transcript accumulation, suggesting that the decreased Gus expression and activity is due to the increased K27ME3 accumulation on the extended region, and possibly due to a higher number of cell division cycles that are needed to dilute the repressive mark. And to understand if this is indeed the case, I took my 6-del line once again with delayed and reduced knuckles expression, the same used in the previous analysis, and crossed it with the PRC2 function mutant, the Phi co-suppression line. And this mutant typically has a continuous phenotype but can produce dramatic phenotype changes, namely stunted growth, loss of apical dominance, curled leaves, typical of PRC2 mutants, early flowering and homeotic conversion of leaves and floral organs into carpel-like structures, as you can see here on the right side compared to the wild type on the left. Next. I found that for 6-del and the Phi mutant background, the panel below here, knuckles expression begins at stage 5 instead of stage 6 compared to 6-del, and the knuckles expression pattern is generally stronger from stages 5 until stage 8. I also quantified knuckles expression and K27ME3 enrichment through qPCR and CHIP respectively, wherein knuckles is significantly higher in 6-del Phi relative to 6-del and lower K27ME3 levels in 6-del Phi relative to 6-del. Suggesting that K27ME3 accumulation is deposited by PRC2 on the extended knuckles coding region in the next. Similarly, I also wanted to find out the role of the cell cycle in this process, particularly the dilution of, sorry about that, the dilution of the repressive mark. So I once again took my 6-del line and crossed it with an accelerated cell cycle mutant, the KRP quintuple mutant line. And this is also the first time knuckles activation will be genetically associated with the cell cycle, because previous work showed this association through exogenous application of cell cycle accelerators and inhibitors, so just through chemical treatments. And this mutant exhibits increased cell size, cell proliferation, and leaf cell size, likely as a result of enhanced CDK activity, which induces more phosphor-RBR1 and upregulation of the E2F pathway. And previous work also showed that the KRP quintuple mutant displays enhanced growth, particularly that of the embryo or seeds. And I found that for 6-del in the KRP background, knuckles expression also begins at stage 5 instead of stage 6 compared with 6-del. And knuckles expression is generally stronger from stages 5 until stage 8. And we also quantified knuckles expression again via qPCR and saw a significant increase in 6-del KRP compared with 6-del, as well as a significant decrease in the repressive K27ME3 methylation marks, which we did via CHIP, suggesting that progression through the cell cycle regulates the dilution of the repressive trimethylation mark. And additionally, I also wanted to find out if knuckles activation in 6-del can be delayed further by inhibiting the cell cycle. So I used a lower temperature condition at 18 degrees C to grow our plants. And to determine if the cell cycle is indeed inhibited or slower at 18 degrees, I checked the transcription of several cell cycle markers, namely the CDK inhibitors, KRP1, KRP2, the Siamese-related proteins SMR1, SMR2, and SMR3, and found that they are significantly upregulated compared to 6-del grown at 22 degrees. I also checked the expression of the core cell cycle marker CYCB1, a regulator of the G2M transition through a CYCB1 YFP reporter line, and found a more prevalent expression at 22 degrees, here top panels A and B, compared to 18 degrees in floral stages 4 to 6, here the panel C and D at the bottom, suggesting cell cycle inhibition at 18 degrees through upregulation of CKIs, leading to decreased cyclin and possibly CDK activity. And I found that for 6-del grown at 18 degrees, here the panel at the bottom, knuckles expression is further delayed, wherein we see induction only at stage 7. I also observed a significant increase in the K27ME3 marks for 6-del grown at 18 degrees, suggesting that the dilution of the repressive marks is delayed due to cell cycle inhibition. And overall, this emphasizes that the cell cycle regulates knuckles chromatin status and transcriptional activation through passive K27ME3 removal or K27ME3 dilution. And to recap and to conclude my talk, we addressed two main questions in our study, namely finding other biotimer genes similar to knuckles and the role of chromatin in this biotimer mechanism with knuckles. And to address our first question, we performed genome-wide in silico analysis and also some implanta analysis and identified 11 candidate biotimer genes, including knuckles. And for our second question, we manipulated knuckles chromatin via adding more del in knuckles. And we found that we can delay knuckles activation this way. And we also investigated the factors surrounding del regulation and found that the repressive K27ME3 mark is deposited on the extended del region via PRC2, and that dilution of the repressive mark on the extended del region is regulated via the cell cycle. Okay. Now, for our future plans, recently we're just mainly trying to get published, and yeah, that's another story altogether, but yeah, which can be described best by me. And we tried to submit initially. Here is a link to the first preprint. If you have some time, please check it out. That one was already declined or rejected, but we also recently submitted to a new journal, and maybe the preprint will come out in a couple of days. So yes, I can also send that, and hopefully you guys have some time to check it out. This preprint is basically the version of my thesis, the one I submitted to the university. The second one has some new data. Some of the data that were included here, like the time course qPCR that I showed earlier, is in the second preprint, and I think the second one is a much better version. So yeah. Okay. So now I think, sorry. So I just want to say a big thank you to, this is our lab in Japan, the Plant Stem Cell Regulation and Floral Patterning Lab. I want to highlight or want to say a big thank you to Ito Sensei, my main sensei, Professor Toshiro Ito, and also Yamaguchi Sensei, Nobutoshi Yamaguchi, who has recently promoted from assistant professor to associate professor. So yeah, they've been very supportive of my studies and my work. And of course, also a shout out to my department here in the Philippines, Ateneo, Department of Biology. I was able to do my PhD through my connection with them. So thank you very much. And of course, to the PSDB for inviting me to give this talk. It's my first time to share my study with a local audience, so it's very special. Thank you to JC, um, Julian Galvez, my friend from DICE. He's also doing his PhD in DICE, who put my name forward to give the talk. I'm not sure if JC is watching, but yeah. Yeah. So yeah, also of course everyone who participated today and who spent their afternoon to watch the talk. So thank you so much. And I look forward to hearing your questions. Okay. I think that's it. Yeah. Thank you.
Thank you very much, Dr. Pelayo. That was a talk, and it was very interesting.
Thank you.
Yeah, I hope you will be around still after Dr. Jobriel will deliver his talk, so you can answer the questions in a little while, okay?
Yes, ma'am.
All right, so we move on to the next speaker. Our next speaker, Mr. Jobriel C. Baluyot, with an MSc in Biology, as I've stated a while ago, is from the Regen Biology Research Laboratory, Institute of Biology, College of Science, University of the Philippines in Diliman. His talk on benchmarking endocrine disruptors in water samples using transcriptionally activated human cell lines. And yeah, Jobriel obtained his bachelor's degree in biology from the University of the Philippines, Baguio, and his master's in biology from the University of the Philippines in Diliman. His research interests include mammalian cell culture, molecular phylogenetics, endocrine disruptors, emerging contaminants, microbial enzymes, bioassays, and gene expression analysis. As part of his master's thesis, he explored the potential of fungal enzymes in the removal of endocrine disruptive chemicals. Currently, Mr. Baluyot is a lead research staff at the Institute of Biology, University of the Philippines, Diliman. He is part of a team currently developing transcriptionally activated human cell lines to trace the presence of emerging contaminants and endocrine disruptors in major exposure routes in the Philippines. Their team is also investigating whether our drinking water and wastewater contain biologically relevant doses of endocrine disruptors that could potentially end up in our body and the environment. So, another very interesting talk coming up from Mr. Jobriel C. Baluyot. My only involvement in the life of Jobriel is that I was once his teacher in UP Diliman. So may I call on Jobriel Baluyot, please, for to take on the stage. Jobriel?
Thank you for the introduction, Ma'am Jacinto. And it has been a while since we have met again because of the pandemic. But I thank you for inviting me again for presenting my talk on endocrine disruptors. So let me just share my screen first. Okay. So before I present my title, I would like to share some items that are generally related to one another in the context of today's talk. First off,